One of Koji's strengths is its ability to identify synthetic genes and genomic regions among related genomes. Here we're going to use a gigantea gene of foxtail millet in order to identify syntalogs in the related genomes of brachydpodium, rice, sorghum, and maize. When these results are returned, we can see that we've identified one syntalog in each of these species, except for maize where we've identified two. We now can analyze these regions in more detail using microsynteny analyses. When the results of these analyses are returned, we can quickly identify that these regions are synthetic based on a collinear pattern of genes. Each one of these panels represents one of these genomic regions, with foxtail millet on the top and zea maize, two regions down here on the bottom. These colored blocks represent regions of sequence similarity, and when we highlight them between regions, we can see the collinear pattern of genes that's indicative of these regions of being synthetic. We can quickly and dynamically adjust these analyses in order to analyze our particular gene of interest in much finer detail. Here, we're analyzing the gigantea gene using a sensitive algorithm, and these colored blocks represent regions of sequence conservation. We can see that they overlap the coding sequences, which are shown either in yellow or green, as well as additional conservation upstream of these genes in the 5' region. These particular sequences may be involved in the regulation of the gene. We can see that some of these have been conserved since the divergences of the brachydpodium and rice lineage from that of maize, and that these would be good targets if we were interested in 6 acting elements affecting the regulation of this particular gene. We can do similar kinds of analyses taking a look at various accessions of maize by stacking all of these on top of one another. Here, our region of sequence similarity is colored so that we can quickly identify those that are common or divergent between these various accessions in order to identify those putative regions that may be regulating differences in gene usage between these particular accessions.